All right, welcome back to the second ever episode of ATTV. I'm Ben McLeod, founder of Convey Bio, and I am here with Stephen Dublin and David Hermanson from Bio-Techne. Gentlemen, really excited to have a conversation with you today about cell therapies.
Yeah, looking forward to it.
Yep. Excellent. All right, so Stephen, we'll start with you.
Yeah.
We've got a pretty big problem inside of the space with regards to high manufacturing failure rates. Yeah. What do we as an industry need to do to help ensure commercial viability? Obviously that's massively increasing costs, but it's also reducing the ability for patients to access these therapies. What do we need to see to change that?
Yeah, it's a meaty question. So always so before I get into my opinion, it's always nice, let's set the common ground, and I think we all agree that advanced therapies hold a lot of promise in the field. Yeah. We all know that manufacturing is a key component of that. I think where things get a little, where the rubber meets the road, where it gets a little bit more tricky, it's almost, it reminds me of that marketing cliche, right? Like no one wants a hammer, they want a nail in a wall, and nobody really wants manufacturing for manufacturing's sake. What you're trying to get is to that first good clinical data. And so I think there's a lot of pressure that's coming from investments, and it's coming from internal pressures as well, is to say, how do I get that first signal, that first efficacy biology data? And there is, let's be honest, we cut corners sometimes, we make sacrifices, and those things will unintentionally set you up sometimes for commercial failures that can either delay your product development, or it can even affect a launch there. So I think what we need to do is stay focused on continuous optimization. We're all looking to how do we get to that first data quickly, but when we're considering manufacturing, and it's not just on the developers but also on the technology providers and the raw materials suppliers, let's work together so that we're doing that continuous optimization.
Excellent, very good. David, if we come over to you now, what strategies do we have to bridge this gap? What should we be looking for in terms of innovation that can help change this?
Yeah, so I think, in terms of strategies that we have to bridge the gap, I think just look around the room here at ATW. We have all kinds of closed systems. I think that we've had a lot of great progress on closing our manufacturing platforms. And some of the innovations around there. Areas that I think we can continue to improve on is when you move into the reagent side of things. So while we have all of these closed manufacturing platforms, some of our selection reagents or cytokines, media is pretty well closed, but a lot of the other reagents that you have to add into the process do not come in a fully closed. And so you still need to go into a hood, you still need to spike that, you need to figure out a way to deliver it. And so I think there's a lot of room for innovation around that would streamline, reduce the amount of aliquoting you need to do of things like cytokines. That's why one of the things that Bio-Techne is showing at this show is our ProPack solution, which is a fully weldable cytokine. But yeah, and I think just all of that to bring all the parts together and not just focus on kind of the big picture, which is always the manufacturing platform, right? Like that's the big thing, that's the instrument. We have those now. I think there's probably 7 to 10 different options that you could go with to achieve closed systems there. But now it's how do we bring the reagents along with it?
Right. So yeah, that really kind of brings up the question of striking that balance, where obviously we need to have closed systems when we're talking about something that's going to be consistently going into patients. But how do we strike that balance in terms of a standardized process on one side, but then also still having something that's a flexible system early, so that you can figure out what's best for your particular therapy and then move it to something that's a bit more standardized.
Yeah, exactly. So obviously you can't start with a closed system. You've got to be able to do your process development. You've got to be able to do your R&D. And so if you try to jump straight into a closed system right away, you're going to burn up too much in cost. You can't run parallel things, right? So it's just too expensive to try to do. So I think really, again, here, this is an area that Bio-Techne spends a lot of time thinking about as a reagent provider. And so some of the ways that we're approaching it is through offering varying offerings of things like cytokines. So we have lyophilized cytokines, that's clearly the standard of the field. We've also done liquid-filled vials, and now we have our ProPacks, which are a fully closed weldable system. And so you can kind of grow as you move through that process. And I think you can apply that to anything, right? That's an example of cytokines. But you can apply that to anything in terms of you got to start out where you can do small scale. This goes to what Stephen was saying. You've got to be able to scale back down, right? So you've got to have a manufacturing platform that allows you to do things, to learn, to do process development, to do things in parallel, and then strike that balance against, okay, once you're in commercial, now you want that more automation and things like that. And I've lost count of the number of times I've already heard automation, and we're only like an hour or two into the day. And so clearly everyone's got to focus on that. But how you define automation also is kind of a big component here. And it's not necessarily just the all-in-one end-to-end solution. It can be a modular approach to automation as well.
Gotcha. So apologies, we're going to talk about automation a little bit more, and I'll take this one to you, Stephen. So obviously with automation, I think it's got a lot of promise inside of the field, but I wouldn't say, it doesn't seem to be fully mainstream yet. Why do you think that is?
No, it's a good question, and it's not a new question too, right? So I can remember 10 years ago being at the very same conference. Facilitate did a great job putting together platforms where we were talking about what's going to set us up for success in the future. Is that fully closed, fully automated system, or are we talking more about really unit operation, modular approaches, and how do we bridge that piece? And after a lot of really deep thoughts and really kind of sometimes heated discussions, but collegial heated discussions, you came down to the answer ultimately was, depends, right? So it depends on biology. It depends on what your investment strategy is. It depends on your facilities, what kind of, your skill set that you have in-house. There's a lot of things there. So, but then the next question came for the people who were saying, these automated systems are really important. The question was, but is the technology we have now going to suit our current challenges? And then will that technology then be applicable into our future needs, right? And that's a big ask to make a technology now that's going to fit in the future. But actually, I think David referenced it earlier. Look around here. You can walk around and see everything from fully enclosed, massively parallel manufacturing with integrated QC, which I don't think anybody was even dreaming of, even 5 years ago, right? And then, now also for as much as we heard automation, we were also hearing AI, machine learning. Like, how do we start integrating that kind of smart data handling in there? So, and then the miniaturization too. So there's some incredible technologies on how do we get things into microfluidics and massively parallel. Mm-hmm. So now, it's kind of like a candy shop for kids at this point, right? It's like we didn't even dream of what, 10 years ago, we weren't even dreaming of what you can just now walk up and just put your hands on. So what's the challenge now? Well, we're still stuck with a big challenge, which is that we've got a lot of variable processes, and then when you start saying, okay, how's company X going to automate 100 different approaches to a manufacturing process, that's a tough ask. And so, one thing that I think you have to focus on, okay, what's the big challenge? And I think what we hear from a lot of customers when we're speaking is that the challenge of, when we were talking about automation, it was a lot about how do we scale up and out? So how do we get bigger, right? And actually the challenge now is okay, kind of harkens back to our early questions. It's how do I optimize an existing process for commercial success? Because people are starting to get that earlier. Let's get this set up earlier. By phase one, let's try to really start getting, what is that final product's going to look like? And so that is not up and out, but it's also how do I circle back and do that optimization at a smaller scale, make those learnings, improve that process, that data, and then scale it back again. So it's an iterative process. And I think that's what I think that's the next horizon of saying,
yeah, definitely. Yeah, and just personally as well, maybe I can pick your brain on this for a second. That integration of automation, which I believe does have true potential. It's certainly coming, but then being able to mix that, I think, with process analytical technologies and then AI kind of built on top, I think is going to even address the closed issue that we were talking about earlier, where if you do have loads of different data that you're learning about your process, that is in somehow getting analyzed by machine learning, and it's able to figure out, okay, based off of the combination of these 10 different variables, it seems that the batch is starting to kind of fail a little bit. Actually, if we microdose with glucose right now, that might be able to fix it. So I'm curious to hear how you think, automation, AI, and process analytical technology can kind of come together to fix that problem.
Yeah, that's a really great point. And it's kind of what we've been talking about too. It's like big data sets, being able to manipulate your process for optimization. It doesn't always go straight up and out. So those kind of alterations that you're saying as well, we've got big data sets and we can learn little things about our process, go back, optimize that, and bring it back down. So when we're talking about big data and machine learning and AI, it can also mean bringing things back to a proof of concept improvement into your process and then back out as well. Yeah, it's a really great point.
Yeah. So even briefly mentioned, microdosing glucose or stuff like that. Obviously, one of the challenges is, finding the right raw materials in the right format and being able to use them in the right context. How can Bio-Techne help with that?
It's a really good question too, because I think that's a really terrific question, especially for companies that are doing raw material supplies. We talk about being partners, and I think we have to be true partners, which means we've got off-the-shelf, vial sizes, for instance. So David was speaking about liquid formats instead of lyophilized formats. That might be more appropriate to how your process is set up and how your team are building your product. And then you go a step further is like building out process-sized vials as well for raw material supplies. Really important. Encapsulating things, putting things in bags. How do we deliver things? We're talking now a lot about T cells and such, but we also have things like regenerative medicine applications. And like, how does your supplier then work to build that, those supplies into your process instead of you trying to retrofit there? So we talk a lot about engineering and instruments, but that raw material supply, you need to have a good partner that understands you and they're willing to work with you, which could be formats. We talk about formats, but it can also be commercial plans, or how do we build a future together as a developer and a supplier, either in raw materials or technology. So look for that when you're looking for raw material suppliers. Really have that in mind. You have to be questioning them. Is this a good partner? Do I trust these people, and do they have the capability to grow with me and adapt with me as much as my maybe cell manufacturing platform would as well.
Makes sense. Excellent. All right, final question. We'll go back to you, David. In the next 3 to 5 years, what do you think we're going to need to see in terms of tools and technology and different solutions inside of the field?
Yeah, I think you guys have already kind of hit on it with the AI theme, right? So I'll kind of just continue on an AI theme here. I think one of the big things is taking all of that analytical data that we are collecting in terms of metabolites or dissolved oxygen or pH throughout the cultures, and then being able to actually act on it, right? And so I think we're sort of in the data set building phase of it, and I look forward to, in the next 3 to 5 years, actually being able to impact and to change the outcome, and take something that maybe would have been an unsuccessful process and change and really go back to our original goal, which is treat patients. Take a patient that wouldn't have been able to successfully be treated with it, and because we've collected all this data, now be able to actually treat them, right? So I think being able to act on it, and just a little bit more of an extension on the AI theme. Some of the other things that we're working on at Bio-Techne is kind of AI modified proteins. And so, can we alter the cytokine stability so that you can avoid redosing? Can we tweak what maybe if it, one of the things that we've looked at is IL-2 alpha versus beta binding. And if we could tweak towards one or the other to maybe help in like a TIL therapy where you don't want to expand the Tregs, right? And so can you bias the way that the cytokines are binding the receptors? And so I think that's going to take a few years to kind of really get going and to be accepted in the field, but it's another area of innovation that I think will really make an impact.
Mm-hmm. Excellent. Yeah. For me personally, I think I'm a big iPSC fan. I just it's just the way my brain goes. So, and it's been so exciting seeing like iPSC derived therapies hitting things with big indications that really affect the population, like neurodegenerative diseases, seeing great clinical trial data starting to come out, and doing things that we could only dream of again, previously. But there are still challenges. So, we have to figure out how to get a lot of these 2D cultures into 3D so that we can really scale up. So we spend a lot of time talking about commercial viability of, CAR Ts, Tregs, TILs. But, it's really going to say, okay, when you start talking about, those things like neurons, iPSC-derived neurons, the sheer quantity of cells that you need as well are really a challenge. And so how do I get it out of, X number of, of flat surface, 2D cultures into a 3D so we can hit those numbers? And then how do we optimize also like those differentiation processes so we can get them shorter to reduce not just the cost, but also the risks involved in a 30, 45 day culture. So really exciting stuff, I think, going forward around regenerative medicine, especially on iPSC-derived things. And don't forget as well, like there's a lot of great work happening on iPSC-derived cell models to help build next generation gene therapies as well. And that's kind of like a unsung hero of that iPSC thing too. They're just quietly doing the work under there, but saying, okay, can I make a cool culture and then see how this drug actually affects it? And can I make a cardiomyocyte and really use that for screening for toxicity and such? So there's good work going on there, but yeah, we need the technology to bring that to the next level. So.
Excellent. Yeah. Very good. Well, gentlemen, thank you very much for coming on. Excited to see where the industry goes in the next couple of years.
Yeah.
Thanks again.
Yeah, great conversation. Thanks.